Hello, everyone. My name is Jim Adelman, and thanks so much for coming to my virtual talk here. And thank you so much to the organizers for including me in a fantastic symposium. So I'm going to talk to you today about behavior of infected hosts, specifically this idea of behavioral tolerance and what that could mean for host competence or the ability to transmit pathogens. And in this crowd, I don't think I really need to tell you that behavior is integral to infectious disease dynamics. It influences transmission. It is influenced by infection. And so I'm going to argue that this concept of tolerance is a great way to measure some important components of transmission, of the behavioral impacts on transmission. But let's start with some definitions first. And first, I want to contrast tolerance of infection in general against resistance. So resistance being just killing pathogens, clearing pathogens. Tolerance being minimizing the fitness effects of a given number of pathogens. And so what that would look like graphically, and these are going to come up, you'd have something, a proxy for host fitness on the y-axis here, pathogen load on the x. If you had two groups that differed in resistance only, you'd see a pattern like this, where this blue group is much better at killing off the pathogens before they reach a high load, but still has the same slope, the same decrement in fitness per unit pathogen. Now, if they differed in tolerance only, what you'd see is different slopes. So the blue animals are more tolerant. They're able to maintain fitness despite similar pathogen loads with the red groups. Now, Dana Hawley and I started thinking about in animals that there's tissue-specific mechanisms that could affect tolerance as well as behavioral ones. And Jess Stevenson and I have been expanding that in a chapter for the book out of this symposium. Now, what we're thinking with tissue-specific tolerance is you have pathology as your proxy for fitness on the y-axis. So animals doing really well at the top, no tissue damage associated. Animals doing poorly, as this finch with severe conjunctivitis, at the bottom of that axis. And so you can use that as a measure of their likely fitness impact based on these tissue-specific mechanisms of minimizing pathology, right? So the blue animals, more tolerant, less pathology for a given pathogen load. On the behavioral side, you'd have fitness-enhancing behaviors, things like foraging, locomotion, mating, these things on the y-axis. Pathogen load again on the x. And so animals that get sick and really reduce those behaviors for a given pathogen load, they would be less tolerant. Those that maintain or even increase those behaviors at a given pathogen load, they would be more tolerant. And the reason these are both interesting to me is that they can have serious implications for host competence or the ability to transmit pathogens. Those could even be opposite. So here I'm suggesting that with pathology, often pathology can be important for getting pathogen out of a host. So your least tolerant individuals might be your most competent, right? Those that have the pathology and the pathogen load. On the other hand, with behavioral tolerance, those animals that keep those behaviors going, they're going to be, for a directly transmitted pathogen, they're going to be more likely to encounter others, so a higher contact rate, so your more tolerant animals are going to be more competent. So this brings me to two big questions that my lab has been focusing on recently. The first being, how do those types of tolerance actually influence competence? Can we measure that? And then how do they vary across individuals and co-vary across individuals, populations, or species? We've been looking at this in house finches infected with their bacterial pathogen Mycoplasma gallisepticum, which jumped from chickens in the early 90s, causes, fair warning, pathology coming up, severe conjunctivitis in the finch hosts. But that gives us a really tractable system because we can measure that external pathology for tissue-specific tolerance. A scale of 3 per eye, so you're going to see axes of 6 in the talk. Also, they do well in captivity. You can experimentally infect them, and you see these typical behavioral changes with infection. This is the same critter before and during infection, so we can get after behavioral tolerance. So with that first question, how do they influence host competence? Well, I'll talk about some work that my grad student, Rachel Rudin, former grad student, she's graduated, did with house finches at Iowa State. So in experimental epidemics, she had 12 pairs, one bird experimentally infected. Everybody was naive going in. Pathogen load measured twice a week, pathology 3 times a week. We used that to measure transmission in the naive member of the pair. And we looked at video with a scan sample every 10 seconds, 10-second window for every 30 seconds, just asking, does the bird feed in that window of these videos? And we calculated slopes for each individual based on their pre-infection levels, which were 0 for pathogen load and 0 for pathology, and their max for pathology and pathogen load. So you have your less and more tolerant individuals there. For behavioral tolerance, we looked at the proportion of those scan samples that the bird was on the feeder at the initial and the day on which that was lowest during infection. And then we coupled that with the initial and the maximum pathogen load for those slopes. Note that there are more robust ways to do this. You need a lot bigger sample sizes, but we discussed that in our chapter, and it's largely based on the Dingemans and Doctorman paper. So what do we predict? Well, from prior work, we know that with more pathology, there's more pathogen that gets onto feeders, and there's more transmission. Feeders serve as important transmission hubs in this system. So with tissue-specific tolerance, we thought that the least tolerant animals would be the most competent. They would transmit the fastest. Behavioral tolerance, we know that more time on feeders means more transmission. So we predicted that the most tolerant individuals would be the ones that transmitted most rapidly. And this is largely what we see. So what I have here is tissue-specific tolerance on the x-axis as a z-score, so we can compare these two, with animals doing worse over on the left and days to transmission on the y. And what you see is more rapid transmission, so higher competence, with those less tolerant individuals. For behavioral tolerance, what you see is faster transmission with your more tolerant individuals. Again, this goes with our predictions. So it looks like in this system, we've got tissue-specific tolerance, decreasing competence, behavioral tolerance, increasing competence. Now, how do those co-vary? Well, let's look at the population level. That's where we have a pretty good amount of data. From a big comparative study we've done recently, now we know that mycoplasma spread from east to west, so that gives us this gradient, and they haven't been to Hawaii yet, that gives us a gradient in time since MG invasion, and we've found mycoplasma invasion, and we found that populations with a longer history show higher tissue-specific tolerance, and that's work with my postdoc Amby Henshin and Dana Hawley at Virginia Tech. So that looks like this. You've got a shallower slope of that I-score versus pathogen load for those populations with a longer history. Then have a grad student, Frank Tillman, who's curious about how does behavioral tolerance vary with endemism. And what he's done is very, very preliminary, very hot off the presses, looked at 2 populations for right now, figured out this great tracking software, did a lot of tweaks to make this work. And what we can get from this is the proportion of time animals spend active, or mobile. So on their least mobile day, that's during the infection there, is on the y-axis and at the peak pathogen load. Now we have a few different time points here. We have pre-infection, right? So that's just all the pre-infection data, no pathogen load, what they're doing regularly, low dose and high dose animals. And what we see is no big pattern here, though I will note that those sample sizes are quite a bit smaller right now than for the other experiment for tissue-specific tolerance. All right, so what can we say about that covariation right now? Well, we've got a cleaner signal for variation in tissue-specific tolerance, but we're in the early days right now, so I can't make too many solid conclusions. But why we care is because tissue-specific and behavioral tolerance can push competence in opposite directions. So if one of those is evolving more rapidly or is more variable, we can better predict the selective pressures that tolerance, broadly writ, places on animal pathogens. All right, with that, I have a lot of folks that I didn't mention in the talk that helped out with the critters, funding sources, and I'm happy to take questions if we have any time. Thanks so much.